general statement that he knows most people here, so most people don't know him. He was working at KSIConnect long ago, and he used to work with me when I was in the chickpea breeding between 82 to 87. So that is the reason why Dr. Sharma said, I need to introduce him rather than him. So we have had a long association, and in the sense that even when he left KSAT and went to do his master's and PhD in Belgium, we still had a contact, and even when he moved to the US, we were still in contact, and we have had lots of research collaboration with him. Almost 10 years. Yeah, almost 10 years. I visited him in 2004 in Penn State, and of course a few others like Dr. H.C. Sharma also visited him. And although he no longer works with chickpea, which is not a good sign for me, I thought he could continue, but he has found greener pastures in maize, but also with KSAT he has been working on sorghum on many aspects. So today he is going to talk to us on plant defense against pathogens and pests through Phytoalexins, and I welcome Surinder now to give his seminar. Thanks, Surinder. So, what we would like you to do is to maybe talk for about 30-35 minutes, Surinder, and then we can open up for discussion.
Thank you. Can you all hear me?
Yeah, come nearer to the microphone. Okay.
Okay.
All of that.
Is it good?
It's okay, it's okay.
All right. I may be moving a little bit, so let me just move this up here. All right. Well, first of all, Dr. Gowda, Dr. Sharma, Hari Sharma, and KK Sharma. Thank you very much, and Srinivas Rao, Rajan Sharma. Thank you for all the collaboration and this invitation to come here and present some of our work that we have been doing last, say, about 14, 15 years ago. We started working on sorghum. Before that, I want to say that when I walked in 304 and 5, the memory of cleaning the gunny bags and removing all my produce, my harvest that I did from the field, those memories came back. And I remember Dr. Gowda and Dr. Jagdish Kumar and myself selecting those chickpeas, selecting those accession lines in the field and moving on with the breeding program. So it was really a very nice, in fact, kind of like a surprise for myself that I could think back and relate again myself to the field aspect that we were doing. Anyway, so last 14, 15 years, my lab has been working on maize, and we started some work on sorghum when I moved to Penn State. Before Penn State, I was working at Iowa State, where I actually picked up the maize. Iowa is in the Midwest, which is the hub for the maize breeding work. So most of the work that we do, in fact, involve several molecular aspects of maize, and also there's a huge sort of field aspect because we grow accessions, we do breeding, and we do molecular type of breeding in order to understand some basic mechanisms as well as take those into application. And I'll explain that to you as we go through the slides. So just an overview of one of the projects that we work on is we try to understand what is the cause of allelic diversity. So we have used a gene known as P1 or pericarp color 1, which produces action of this gene is a transcription factor. It produces these compounds known as phhlobaphenes. So these are the red pigments on the pericarp. So there are about over 100 alleles of this gene, and this has been puzzling to maize geneticists that why there are so many alleles. I won't be talking to you about this project because this is solely a maize project, and I'm here as a sorghum person, not as a maize person. So anyway, so the goal is to identify what are the mechanisms that control allelic diversity. Why there are 100 alleles at the same locus. And it turns out that epigenetics, which is DNA methylation, chromatin modification, these are the ones that are responsible mainly together with the gene structure, how the structure has evolved and how the evolution of the structure led to the epigenetic modification. In fact, with us today we have my friend and colleague who visited us as a Borlaug fellow. He came all the way from Dr. Sanjeev Deshpande. He actually worked with us on this project. So he visited us a few months ago as a Borlaug fellow. So the work that I'm now presenting here, which we are focusing on, is the plant defense compounds, and we are mainly interested in these compounds that are known as flavonoids. So flavonoids, you all know, there are these flavonoids in the fruit, and there are flavonoids in the tea, in the green tea, in the chocolate. And then you have the flavonoids that are giving the beautiful colors to the flowers. So you can, so the pollinators can get attracted and participate in pollination. So what we are interested in, this group of these compounds, which are basically derivatives of or derived from the basic structure of flavonoids. Flavonoid structure is a 3-ring structure. Basically, when you see 3 rings in any biochemistry textbook or something, so these are the derivatives of flavonoids. Okay, so in sorghum, we got interested in the role of flavonoids, the role of these compounds as antifungal compounds as well as insecticidal compounds. And I'll talk to you more about those. So here's a sort of brief outline of the work that we're presenting now. So our model system is Colletotrichum, the anthracnose causing fungus. And then so we are testing several. So my lab is a genetics lab, and everything that we do, we base it on the phenotype as well as the genotype. So our goal is to produce a lot of mutations, knock off the function of the gene, and then ask the question if that knocked out gene controls the pathway that we are interested in. Okay, so these are the compounds that I'll be talking to, and they are also referred to in the literature as phytoalexins. So phytoalexin is a, sort of like a general term that any compound that can ward off the microorganisms will be called phytoalexins. So I'll be saying that the term phytoalexin or antifungal or insecticidal compounds. So my lab is also heavily involved in developing sort of genetic resources in terms of fungal strains that are tagged with different GFPs. Also the development of the transposon tagging lines in the grain sweet sorghum as well as in the forage sorghum. And also I'll give you a little overview of that. And at the end, I'll give you a flavor of some of the work that we have started on sorghum insect interaction, and that has brought us now much closer to Dr. H.C. Sharma, and we are starting this new collaboration soon. Okay, so anthracnose and sorghum, I mean, there are many people here that work on anthracnose, so I don't want to go into too much details. Rajan Sharma has been collaborating with us, working on one of the USDA and NIFA project. We're trying to understand how to produce anthracnose resistant lines for climates like in Pennsylvania, which is high altitude area. Okay, so however, this work actually started a long time ago, and this is one of the famous publication from Ralph Nicholson, Snyder and Nicholson published in 1990 in Science, that when a conidia or the spore of this fungus lands on the sorghum epidermal or the leaf of the sorghum epidermis of the sorghum leaf, then the plant actually produces within 24 to 36 hours after infection, it produces these small lesions, and if you look at these lesions very closely under microscope, you will see that there are inclusion bodies, or there are bodies that contain these compounds, and these compounds then approach the primary site of infection, coalesce together, and then release the compound. And these released compounds then challenge the cell and the fungus which is present in that cell and kills the fungus. So this has been a very classical work done by Nicholson at Purdue University, late Professor Nicholson. So what you see in the field, these reddish brown pigments starting with the small lesions, also called, that's why anthracnose is called like coal, because you see this coal-like symptoms. And then, of course, your leaf can indicate like a blight kind of phenotype. Anthracnose leaf blight is commonly called. Okay, so this was already known that this is sort of like the infection pathway. And what my lab is interested on is that what are the genes or what is the genetics of the plant that require production of these antifungal compounds, because these compounds are not constitutive, they are induced. So because they are not constituted, they're induced, so it allows us to find out, so what are the elicitors required for the induction, okay? So what are the fungal proteins that are required for the induction of these compounds? So that's something we're doing. But before that, I want to give you a little overview of the maize and sorghum world of pigments. Okay, so there are two types of pigments in maize and sorghum. One is this commonly known as anthocyanins. So anthocyanins are red or purple pigments. You see them on the endosperm, and also you see them on the plant body. But then there is a common, or rather less common pigment, is this one called Phlobaphenes. Phlobaphenes are not in the endosperm or not in the aileron, but they are in the pericarp. Pericarp is the outermost layer of the kernel. These pigments are also present in the glume, which is the palea and lemma, the botanical structures of the plant, the botanical names for these glumes. So so we have developed over the years genotypes that would show you a phenotype like this, which is red pericarp and red cob. Similarly, in sorghum, you have the same nomenclature we have used, where we have the Y1-RR, means there is a red pericarp and red inner glumes. That is a phenotype. But interestingly, what is more interesting is the evolutionary aspect, that in maize, these Phhlobaphenes are produced in the silk and not in the leaves, whereas in sorghum, these compounds are produced in the pericarp, glumes, as well as in the leaf. So there is an implication of this in the anthracnose resistance. Later I will talk about that. Okay, so so I told you about the anthocyanin pathway, and then you have the Phlobaphene pathway. Precursor of this Phlobapene pathway is luteophorol. Okay, luteophorol, which converts into Phlobaphens by this polymerization reaction, and then you have the formation of the antifungal compounds in sorghum leaves, which are called luteolinidin and epigenidin. Okay, so I'll be focusing more on the luteolinidin and epigenidin part, while talking a little bit about the precursor, which is the luteophorol. So precursor is present both in sorghum and in maize and sorghum, but maize only produces these compounds in the silk as well as these floral tissue. Sorghum produces, in addition to this, it produces these antifungal compounds in these structures. Okay, so one of the first things that we did, asked the question that if there is a homologue of P1, so P1 is the pericarp color one in maize. So we isolated the homologue of that in sorghum. So if sorghum homologue, known as the Y1 or the yellow seed one, is required for production of these pigments, then when we have a transposon knocking it out, then we should see this candy stripe phenotype. So in fact, we were fortunate to find this genetic stock from Sudan. In fact, this was isolated by late Professor John Axtell at Purdue University. He found this line and he gave it to me and said, hey, we want to isolate this transposon because this is the time of 1980s when transposons were very popular. So the work on transposon tagging and all that was becoming popular at that time. So what we did, we isolated this transposon as well as along with the transposon, we studied the structure of this Y1 gene. Now in a few minutes I'll tell you that, so Y1 is a homolog of the Maize P1 gene. And I told you that in the Maize P1 gene does not produce pigmentation in the leaves, but only the Y1 gene produces pigmentation in the leaf. So the reason for that is that the promoters of these two genes are highly diverse. So because of the diversity in the promoter of cis-regulatory elements, they are causing this induction of these compounds, which are more sort of like wound-inducible compounds or fungal challenge compounds. So we use a very simple assay in the lab. We adapted that from Ralph Nicholson's lab. So you have the seeds, you germinated them in the dark, so you produce etiolated seedlings. You have the long elongated mesocardos. You inoculate that with the fungus. We use two different fungi. One is Cochleobolus heterostrophus, which is actually non-pathogenic to sorghum, but it produces the same symptoms of production of these phytoalexins. So we also use Colletotrichum graminicola, but the Colletotrichum sublineolum, excuse me. So the reaction that is caused by Cochleobolus heterostrophus is much faster. So within 24 hours, you can start to see these compounds against the white background, whereas the Colletotrichum sublineolum takes about 1 week to 10 days to see this sort of phenotype. So that's why we use this non-pathogenic fungus. Okay, so once we isolated this stalk, from the stalk in which the transposon was inserted in the Y gene, by simple genetics and breeding and molecular work, we were able to isolate the revertant lines in which the transposon has left, as well as the null lines in which the transposon actually has created large deletions. So when you compare these two near isogenic lines for the fungal inoculated challenge, you can see that then the lines that have the reversion phenotype of the Y1 is also producing the phytoalexin compounds, whereas the mutant lines like Y1WW1 here indicated as a recessive mutation because of the loss of function, this line does not produce these compounds. So we routinely we can measure these compounds. These are by HPLC, luteolinidin and epigenidin. They have a retention times of about 18 to 19 minutes after you load the HPLC. Similarly, these compounds can be then separated on a mass spec, and the resistant line produce abundant compounds, whereas the susceptible, or I shouldn't say susceptible yet, because the null line does not produce these compounds. Majority of those compounds. So then several years ago, then Aoife Ghafoor joined as a postdoc, and she did the work on these mutants to see the infection severity of these mutant lines, the WW1 and 4, compare that with the revertant lines, and found that the lines that do not produce these phytoalexins, they are actually susceptible to the fungus. So Aoife has also developed GFP tag lines. The goal here is to go a little bit more deeper into the upstream region of the pathway to find out that what sort of signaling is involved in anthracnose sorghum interaction, because that's one area that's not really known to anyone. Okay, so in 2010, in fact, Dr. Thakur at KSIConnect and Dr. Reddy and Dr. Gowda, I discussed with them some of the grant proposal funding that were available for the Bioenergy program. So we came together, and now we are working with P. Srinivas Rao and Rajan Sharma, and our goal here is to identify sorghum lines that, when are grown in Pennsylvania, and of course here, those lines have, when the debris of those lines fall in the ground, they produce, they allow poor production of acerbi. So what I'm trying to say is that when the fungus overwinters in soil, the overwintered conidia then go back and attack the next generation of the plant. Okay, both sorghum as well as maize. And in Pennsylvania we do rotations of maize, soybean, and sometimes farmers grow sorghum, but that's only in the marginal land. Okay, so because of the bioenergy push, there is again, farmers are interested in growing alternate sorghum crop for alternate bioenergy instead of throwing out the maize for the bioenergy program. In fact, they are trying to grow the sorghum. But because sorghum is a newly introduced crop, there are new diseases and insect pests that are being introduced. So the goal here of this project is to find lines that can sustain with least amount of fungicide on one hand, and also least amount of the infection by these anthracnose fungus. So the project has involved a heavy outreach program. So here we see some of the scientists and the farmers talking to IFA, and Srinivas visited us last year, I think, and Sanjeev was also there at that time. So they're evaluating this anthracnose disease nursery in one of the trial. One of the graduate students is doing selections. So this project has been going really well. So this is a 5 years project. So we hope to get some interesting aspects of this project, which are listed here actually to be done in the next 2 to 3 years. So we are focused on isolating several strains of Colletotrichum. We have also screened several sweet sorghum lines using these 11 new isolates of sublineolum. Then field trials are in progress. Nested association mapping population has been used, and we are going to use we're starting to use the RNA-seq results in order to go into the identification of candidate genes. Then ICRISAT, with this collaboration project, ICRISAT has already screened 500 or so lines and came down to 28 highly resistant lines that we are testing now in the greenhouse at the Penn State greenhouse. But once those tests will be over, then those lines will be transferred to the field. Okay, so then transposon induced lines, I'll talk to you a little bit in a few minutes. Then, of course, we are trying to find cultivars that, when are grown, so that the overwintering is avoided. Overwintering of the fungus is avoided, those strains. And the reason, the hypothesis behind is that if a sorghum line is producing antifungal compounds, then these antifungal compounds should provide resistance even when you have those debris or stock as a debris during the winter season. That's our sort of hypothesis, which is kind of a revolutionary idea, but we are testing that. We already tested 3 years of winter-summer cycle, and it seems that there is a activity of these antifungal compounds in the soil when you are left out with the debris. So just an overview that we're testing several near isogenic lines. Either they are developed by our program or USDA ARS developed those. Similarly, this is a typical sort of phenotype. When you see a resistant line or versus you see a susceptible line. So there is a clear sort of resistant, susceptible phenotype that you can observe. And then once you observe those areas of susceptibility, you can take those and do the HPLC and find if those lines contain the compounds. I'll give you a little better picture of that. So for example, here is a set of resistant lines, and here is a set of susceptible lines. So the resistant line produce these compounds, whereas susceptible lines don't produce these compounds after inoculating with the fungus. So as I said that we are interested in the signaling pathway, and our collaborator in this project, the USDA funded project, Lisa Valencourt, she's a fungal biologist, and she has isolated several receptor proteins, several elicitors from the fungus, and we're using that in order to identify the receptor proteins in sorghum. Because the pathway is very simple, that you have the receptor elicitor interaction, and then this interaction gives you the signal. The signal somehow, one of the components of the signal is a Y1 gene, Y1 is a transcription factor, it produces these flavonoid compounds. Flavonoid compounds are transported on the ER, endoplasmic reticulum, and then they are moved towards in the vesicles, they move towards the primary site of infection. So developing some of the genetic resources, because sorghum is one crop that has lagged in terms of the transposon-based tagging. So sorghum actually has no transposon tagging tool. So my lab actually is trying to develop this transposon tagging tool. We're starting off with the candy stripe line. Candy stripe line has a transposon in the Y1 gene in the intron. When the transposon leaves, it does not affect the intron, so that the gene can transcribe now and produce a full red head, whereas the transposon can move into any other gene. Okay, so then we can select based on the greenhouse studies as well as in the field, we can select new mutants. Okay, so the one of the first focus is to isolate these mutants that are participating in the production of these phytoalexins, 3-deoxyanthocyanin phytoalexins, which have a role in the resistance to anthracnose. So we are isolating both resistant as well as susceptible mutants. So here is just one example. The transposon in fact can move out and go to a linked location, but also the transposon can go into the unlinked location, can also go into the same linked location. Here is one example that this stalk contains two transposons. One is the original copy as well as the second transposed copy. So this is a very active transposon in sorghum, and it's kind of like the same transposon that Barbara McClintock worked in maize. It's known as ENSPM. Oh, this work was published a couple of years ago, so that's why I didn't want to go into details. So now we have developed very systematically all the stocks that are actually available. If I was talking to Srinivas that we can provide these stocks, and transposon can then be mobilized into forage sorghum as well as sweet sorghum, because this sorghum line is only a grain sorghum line. So there is a lot to be done, for example, in terms of the biomass work or in terms of the sugar metabolism in sorghum. There's not much known, so that can be done by. We also use, when we isolate these new mutants, we compare the wild type and the mutant profile for different compounds. Here is just an example of the phenotypes. So for example, there are phenotypic mutants isolated. It's also published, so not going into much details of all the mutants, but this is one that's really close to my heart, is a mutant that does not produce the bloom, the waxy coat does not produce that, and so we are currently cloning the gene responsible for the bloom. The implication is that, so you have the bloom in sorghum, that's why sorghum provides tolerance to drought. Because I'm a maize geneticist, majority of my duties are in maize, and so the question that we are asking is that why maize do not produce these compounds, these antifungal compounds? So can we engineer these compounds in maize based on the sorghum work? And we found early on that the two transcription factors, the maize P1 and the sorghum Y1, they differ in their expression patterns. Maize P1 has its expression only in the floral tissues, whereas the sorghum Y1 has its expression both in the floral as well as in the vegetative tissues. But on top of that, the sorghum transcription factor can be induced in the presence of stress. It could be a fungal stress, it could be insect stress, or it could be a wounding stress. So there's a big difference between the promoters of these homologs in sorghum and maize. So there are many examples, in fact, when you compare the homologs of sorghum and maize, there are many changes in the promoter cis elements that has caused these variations in expression. So we are working a little bit on producing this transgenic maize, in which we are introducing the whole Y1 gene and producing different patterns of phenotypes, as well as looking at the HPLC profile of these phytoalexins. So it's clear that these compounds are induced, can be induced in maize. The only thing is that this is a more sort of like a basic understanding work. It's difficult to release these into the environment, okay, unless you tag with some company and sell them the license. They will buy the license from you. because in most of U.S. universities, we are not really releasing any cultivars. However, the time is coming that we will start releasing cultivars. So maybe someday that these lines will be important. So anyway, it's just a sort of overview of the HPLC work from the transgenic lines, showing that these lines are producing these antifungal compounds that are important for the anthracnose resistance. All right, so I hope I have like 10 more minutes. So just gonna briefly touch this work on sorghum-aphid interaction. So what we have done is there was an observation a few years ago when we were growing these sorghum mutant and sorghum wild type plants. So when I say to mutant, mutant means the Y1 mutants which have the deletion of the Y1 gene. Okay, so when we're growing these mutants in the wild type revertant plants in the greenhouse, we observed that these mutant plants actually, the aphids are more attracted towards these mutant plants. So my postdoc Rupesh, he started working on this idea that maybe there is something different in these Y1 mutants compared to the wild type plants. But what is the difference between them? So we first set up a simple experiment. Okay, so what he did, so he used 3 near isogenic lines. So these isogenic lines, 2 of them are mutants for the Y1, which means the Y1 is deleted, and one of them is a Y1 wild type or the revertant line. I told you that Y1RR and Y1WW lines. He also included the very famous line which has been sequenced, BTX623. Okay. He used these clip cages, and he released in the clip cages. He put these cages on the alternate leaves, as well as younger leaves and the older leaves. Okay. In each cage, he released 2 mature aphids. Now, I'm not an entomologist, so but I can try to answer questions how Rupesh has actually trained me on this. So he replicated this 3 times. What he found that adult aphids, when adult aphids were removed, he found that 5 immature aphids per cage were left to develop. And what he found is the result. So in the Y1 WW4, so we use both the mutants, Y1 WW1 and 4. In Y1 WW4, the number of aphids was significantly higher as compared to the wild type. And similarly, the BTX control, the number of aphids were less. So it looks like there is some kind of interaction or attraction that aphids were found to develop, better on this Y1WW4 line. He also found that both young as well as the old plants, there was a significant difference as compared to the Y1WW4 and BTX623. Now, when he counted the mean number of aphids left growing in the cage or multiplied in the cage, so the mutant one actually had more number as compared to the 2 wild types. And if you can see them here, they're actually visible here. These aphids are more in the mutant as compared to the wild type. Here is actually the data. In Y1WW4, mean number of aphids significantly different from the Y1RR3 as well as from the BTX623. So he also did some sort of choice test, but he found that by doing the choice test, there were aphids moving towards the Y1WW and not towards the Y1RR. There is a little summary of these preliminary. Again, I would say that these are very preliminary results, and I talked to Dr. Sharma about these results, and so we're going to be testing or confirming these results in more details. Okay, but the preliminary data says that aphids in cages reproduce significantly higher in Y1WW mutant and BTX623 as compared to the wild type or the revertant gene stock. Okay, so in the greenhouse, aphids instead of higher for Y1WWs than the Y1RR3 plants. When plants were subjected to EPG analysis. Now, this is something that is interesting and intriguing. When we did this, so the aphids are ones which feed on the phloem. Okay, so you can do this EPG analysis, and there was no difference observed in the feeding behavior of the aphids for all the 3 lines. So interestingly, what we did, we made artificial diet from these 3 different lines, and we found that so we did a 10x dilution factor, that the Y1RR3 and Y1WW extract, there was significant difference on Y1RR3 compared to the control Y1WW plant diet. So that indicates that there is something going on with these mutant plants. We don't know what exactly is the cause that the aphids are attracted towards that. One possibility could be the volatiles. Okay, that's what Dr. Sharma and I were talking about that. So he has the setup GCMS, so we can try to find out if this is the cause that volatiles are produced in the mutant plant as compared to the wild type plant, so that the aphids are attracted towards those volatiles. And of course, one can do phytohormone analysis and do a little bit better choice tests. That's about what I wanted to talk about, and I really want to thank several people here. These postdoc Aoife and Rupesh significantly contribute to this work. By the way, the new student Dinakaran Ilango is one of the ICRISAT or ex-employee of ICRISAT, I would say. He's just joined me this August, and he's starting his PhD now. So he'll be working on the sorghum, anthracnose and sorghum, excuse me, aphid interaction project with us in this collaborative project. There are several collaborators. The main contributors are here from ICRISAT. Also, these folks here have been contributing in this USDA funded project. And then there are current undergraduate students who are part of this project. Of course, can't thank enough the funding obtained from USDA, NIFA, NSF, USAID, and ICRISAT. Thank you very much for your attention. I'll be happy to take questions.
You want to ask questions? Okay. Thanks, Surinder. Maybe if there are any questions, comments, I can ask people to raise them.
Sure. Thanks for a nice presentation.
Thank you.
These promoters you were talking about, especially from SBY1 and transcription factors, what are the implications? Can they be used for other things also?
Yeah, they are actually can be used for wound-inducible response. Yeah. In fact, what we are doing is, so we have taken the Y1 promoter and developed a cell line with the Gus gene, okay, Gus marker. Excuse me. So the Y1 Gus transgenic cell line is one sort of our planned model. But then what we have done is, from the fungal side, we have isolated the elicitor. So the fungal spores, when you crush them and you filter them, and you take that filtrate and you apply that on the plant, it will produce the same anthracnose phenotype. So it means there is a elicitor, there is a protein in the fungus that can induce the promoter. So this is just a model system that we have developed. You take an elicitor, you take a cell line, and you can induce the Gus gene. Okay. Now the same cell line, we are thinking to use that for many other stress processes. The reason for that is that because this promoter, when you look at the the cis elements of this promoter, they are all kinds of cis elements. They are WD binding proteins. There are wound-inducible wound-inducible cis elements, and then you have fungal-induced cis elements. So there's like a whole battery of cis elements. So in fact, our current goal is to introduce this promoter and Gus cassette into soybean. Excuse me. So one of the things we're trying to do is produce lots of these antifungal compounds. in sorghum. But there's a little bit of problem that you need a huge amount of tissue to extract those compounds. And we found a way out that if you produce hairy root in soybean, right, and then you introduce in the hairy root this Y1 gene, then you can actually modify the isoflavonoid pathway of the soybeans, and you can produce those antifungal compounds in soybean. So that's our current sort of working experiment. Things are going on that our goal is to produce like a huge amount of these compounds and test them then on the plant. In the field, for example, we can spray that, in the field we can spray, instead of spraying a fungicide, we'll be spraying a natural flavonoid compound. It's kind of like in a nutshell what we're thinking.
In your candy stripe plants, do the aphids prefer the white sectors of the product sector?
That's a good question. We really wish that we could see that phenotype. The problem is that the candy stripe phenotype on the leaf does not show up until the very late in the maturity. Okay, and by that time, actually, the aphids are moving around, and but when you have the adult plant, they really don't care what is inhibiting them. So we really want to see that. So one thing we were trying to see if, so we can take a picture of the green plant or the leaf, and then remove the aphids after taking the picture where the aphids are, and then bleach the leaf and see if in the bleached leaf, in the bleached young leaf, we can find those stripes back. So we haven't really done that, but this is something that we're thinking. That will be the proof of the sort of if these compounds are actually playing a role. So, I mean, it looks to me that it's not only the compounds that are because of which the aphids are avoiding the Y1RR plants. It could be something else that we have modified by modifying the pathway by removing the Y1 gene. Maybe those Y1WW mutants are producing something else that aphids are actually more attracted towards those plants. So that's a great question.
So could I, or is there a difference in the structure of the epidermal cells in the Y1 mutant plants? Because I've forgotten what Y1 is, but if many of these transcription factors are specific for epidermal tissues.
Yeah, I understand. It's another great question because so Y1 is a MIB type of transcription factor, and in Arabidopsis, for example, and also in maize, these MIB genes have been implicated in the trichomes, development of trichomes. So we have looked at the surfaces of these Y1WW and RR mutants. There is really no difference in the trichome density, or so there's not really any physical change in their epidermal cells.
Okay. Okay, Rajan.
You have been mainly discussing phytoalexins, right? All the antifungal compounds. And you are also interested in identifying some of the lines that promote the over-seasoning of the pathogen.
Right.
Right. So do you see the role of phytoalexins in preventing the over-seasoning, or it could be some other phytoanticyclines or other antifungal compounds? Because
Yes.
Phytoalexins are induced antifungal compounds.
That's possible. That there's an interference, from other compounds. That's why when we have these mutants versus the isogenic reverted lines, so these lines don't really produce anything else. They're very clean. They're very sort of pure material in the sense that they're only producing these flavonoid compounds, and they have the same background profile of other compounds. That's our assumption, of course.
In that sense, it will be phytoanticyclines rather than phytoalexins.
Yeah, because they're already induced. If they're already induced, they're pre-induced, then it will be, you can call them by that name. Yeah, I think it's just a question of nomenclature. Yeah. But I think this question is good that if they're already induced, so they are sitting there, Yeah, exactly.
In sorghum, there is a huge activity of these phenyl propanoid biosynthetic pathway. There is a set of compounds that are constitutively produced, and their amounts do not change in response to stress, that would be biotic or abiotic, insect, pathogenic, or drought. There is another set of compounds whose amounts increase very rapidly in response to damage by insect or plant. And there are some compounds which almost disappear following insect damage. So this is based on work of fruit fly. So I think you have to look at a range of these compounds that are produced. And in that there are again two sets of compounds. One are the phenolic or flavonoids, and another is the simple carbon chain experiments in the leaf surface bags of sorghum.
Great.
Many of them play an important role in attraction of the insects and or allowing the pathogen to germinate and sporulate. And some of them are very simple compounds like para-hydroxybenzoate and benzaldehyde, which are present about anywhere from 5 to about even 20% of the leaf surface wax amount. These compounds are attractive, say, to the sorghum fruit fly as a oviposition stimulants.
Right.
And some of these, in addition, there are a huge number of another compounds which are simple alcohols, aldehydes, and acids that attract and or repel the insects. So probably we have to look at the whole
Yeah.
chain. So
Yeah.
that's just.
No, I think it's a good comment, and what happens is that with your GCMS, I think we should be able to profile every sort of mutant and the wild type plant, not only just these 3 lines. This is just one example that, so we can, if you can profile those, and that will give us a much better idea between the differences, similarities in these lines. Thank you.
So what is the stability of what is the stability of these genes in the already developed transgenes?
The stability of the
Stability of
transgene.
phytoalexins genes in your already developed transgenes. Did you ever check it?
If I understand correctly, you mean that when we introduce a transgene
Yeah.
to a, let's say in our case, we introduce a maize plant, if that transgene is stable or unstable.
Stable for how long?
Yeah,
How
so
many generations?
it's been now about F8 or F8 and F10 generation, and we haven't seen any silencing. Right, and when we selected these lines, so we have about six independent events of these transgenic lines. 6 independent events came from about 20 total events. That means the 14 events was silent along the way at some point. So we have been, discarding them. Because we are not interested in silencing in this project. We're more interested in, the expression of that, right? But we have another project I mentioned to you, that the epigenetic project, where we are using the silence lines to ask the question, what is the cause of the silencing? And this is not us only. I mean, it's known in the literature that silencing can be caused by multiple copies, right? So there's an instability because of multi-copy transgenes, or there's an instability because of production of small RNAs, for example, right? But we are not really, so when we see a silenced transgene, we don't follow that, because our goal is to keep that expressing. But good question, yeah. Silencing is a problem, especially in the genetically modified plant. In fact, if you talk to the industry people, because industry never talk about silencing, right? So they have all the transgene out there, they are very well expressed, under multiple promoters or dual promoters. So as soon as they see a silence, they will remove it. They won't even come to the, founder seed stage. So we actually I work with people from DuPont, and their main goal is to keep expressing, not silencing.
There are 2 people, the last 2, Julie and Julie.
So there's an unpigmented locus in maize, W1, and so I just wondered if there's an equivalent, the equivalent locus is known as sorghum, and whether it has an effect on aphids. I know it's to do with anthracnose rather than phlobaphene, but
It does not have effect, you see?
it does it. Is the equivalent to the W1 locus of maize known as sorghum?
Okay.
Uh,
W1.
W1, which is just an unpigmented
Okay.
genotype of maize.
Mm-hmm.
So there's W1, W2, 3, 4.
Mm-hmm.
So I take it that the equivalent locus of sorghum isn't known. Is that right?
No, it's there. The problem with sorghum is that the genetics is very poorly known. So, I haven't looked at the particularly W locus, but of course one can, now we have the sequence available for sorghum BTX623. I haven't done a blast of that genome. My thinking would be that it should be there. Most loci that you see in maize, they are quite well preserved in terms of their coding region, very well preserved. The only differences we see between sorghum and maize is in the upstream and downstream regulatory regions. Thank you.
Regarding fungal transformation, what is the methodology you have successfully used for raising GFP constructs of your fungal strains? Is it bacterial mediated, agrobacterial mediated, or any other biological method? What is the methodology you have adopted?
Yeah, it's actually, we use both. More successful actually comes with bialistic bombardment. It's much more simpler, and you can easily easily make transgenic fungal strains with different GFP strains. Yeah. In fact, this is actually a collaboration with a plant pathologist at Penn State, Su Chung Kang, who has actually, with my postdoc IFA, they have developed that.
Yeah, actually, I have a couple of questions. Maybe I'll get in touch with the EFA.
Yeah, sure. Yeah, please. IFA will be here in March.
Oh,
You can get in touch with Dr. Srinivas Rao. He's hosting IFA's visit for this conference.
okay.
So she's actually the fungal biologist working in this project.
Thank you.
Thanks, Surinder, for the nice presentation. I have only a comment and suggestion because seeing on the collaborators list, Jeff Peterson and Scott Settler, they have developed this one near isogenic lines for mutant edits of lignin lines. So both CAD as well as COMT, and in fact some of them we are having. So because Dr. Hari Sharma and you are working on epid interaction, and so lignin is also a product of downstream product of the phenol propionate pathway. There are 3 different types of lignin. So because the near isogenic lines are available, it may be prudent to test their this one interaction with the apids because they have different levels of those lignin content, and we can in fact estimate the lignin content, and we can relate whether that have any relevance to the aphid susceptibility or resistance.
It's good suggestion. Yes. Yeah. Well, actually, Scott has looked at anthracnose in these lines, and he hasn't really found difference. So his result is that there's no correlation in the lignin content or the type of lignin with the anthracnose susceptibility. But I think they haven't probably done yet, or they're doing the insect part. Yeah. But yeah, we can test it with the aphid, interaction.
Currently,
If those lines are available, do you have those lines, you say?
yeah, I have some of those lines.
Yeah.
In fact, got from Jeff. He
Okay.
was working at Lincoln.
Okay.
Now he is, I think, retired.
Retired, yeah. Good. Thank you.
I think it's coming to close. It's 1 minute to 12. So let us thank Professor Chopra, the usual way. Thank you very much.